Hi everyone, I am Emad Magdy, lecturer of nephrology, Mansoura University, and my topic today will be the water sodium potassium control from the interconnected pathways to the clinical relevance. My agenda will be divided into three main sections. Section number 1, some facts about the water itself, the body water content and distribution, the terminology of solutes, electrolytes, tonicity, osmolarity, and at the end how the water follows sodium. Section 2 will concentrate on the multiple systems which control the water, sodium, and potassium homeostasis. The main will be the endocrine system and the kidney, but a very important also role to the GIT and the transmembrane shafts through the cell membrane. Section 3 will concentrate on some issues of applied aspects to the facts discussed in section 1 and 2. How can we think in a case presented with hyponatremia or hypernatremia? What are the applied knowledge of the diuretics? How can the diuretics affect the sodium, potassium, and water homeostasis, and the brain changes in different cases of sodium correction, either hyponatremia or hypernatremia. To start with section 1, the water itself. The water represents around 50 to 70% of our human body. This percentage differs according to the age and according to the gender. So the babies have the largest water content. Nearly 75 or 80% of their bodies are made of water. In females, 50% or 55%, in males, 60%. Why they differ in the water content? Because the water content is related to the body fat content. Since the body fat is practically water free, this means that the female will have less water than the male, because it's supposed to have more fat, and the babies have much less fat, so more water content. It's nice to know that the most water replete organism is the lettuce, with around 95% of the lettuce of this plant of water, while we humans are comparable to the banana. Obesity can be a cause of reduced total body water, and this carry some risks which are related to the obesity, because we know that the water diminish some risks and diminish the headaches and the muscle aches and make the liability to form a kidney stones less and less. So the water, as we know, is a very vital to us, and they have many functions, many protective functions which we like to enhance and increase. Okay, so most of our bodies are of water. What's the constituents in the water? Is it a pure water? It's not a pure water. It's a mixture of cells, proteins, glucose, lipoproteins, electrolytes, and other substances. And this is the time to know some terminologies. What is the terminology of solute? What is a solute? The solute refer to all the dissolved substances in any fluid, but our fluid today is the water, which may be charged, such as sodium, or uncharged, such as glucose. So what is the electrolytes? Electrolytes are substances when dissolved in the water dissociate into charged ions. Some of them will be positive charged, will named cations like the sodium, and the others are negative charged, and will be named anions like the chloride. This is the fact number 1. So that the water is not a pure water. Fact number 2. In human body, the water are distributed in compartments. We are not balloons. We are not water balloons. We have certain compartmental distribution of the water. And the water is distributed into 2 main compartments, the intracellular compartment and the extracellular compartment. The intracellular compartment contains around 60% or 65% of the total body water, and the extracellular compartment represents the remaining third, and this remaining third is divided into around 3/4 in the interstitial space and 1/4 in the plasma. The composition of solutes which dissociate in the water differ between the fluid compartments. We have solutes that is usually present inside the cell and solutes present outside the cell. So we have solutes which is common in the intracellular compartment, like the potassium, and you have solutes which is common in the extracellular compartment, like the sodium. So one of the essential homeostatic functions of the cell or of the body to maintain this compartmental distribution of water and solids, and this is called the osmoregulation. The osmoregulation is made by multiple systems we will discuss in detail in the second section, but I'd like here to highlight what's the meaning of osmoregulation. What's the meaning of osmoregulation? The osmoregulation is the process aiming to maintain the compartmental distribution of water at this between the intracellular and extracellular, and also maintain the compartmental distribution of solutes. Some solutes are abundant inside the cell and the other are abundant outside the cell. This is the cellular targets, and this is the osmoregulation. Fact number 3. Water is never transported actively. It never takes energy to move between compartments. So this is fact number 3. Fact number 4. Cells do not directly control water movement. They do control movement of electrolytes and other solutes, and those electrolytes and solutes have the ability to drag water behind them. So the cell itself cannot drag the water, but the movement of solutes within and within the cell or away from the cell is the force that drag the water inside the cell or outside the cell. So the osmoregulation is the function, as we said before, the osmoregulation's function related with keeping the solutes in balance between or in certain composition inside the cell and outside the cell, and the fluid inside the cell and outside the cell. So it seems that the cell membrane should have a strict function to do this, or have tools to do this. How can the cell achieve its target? It has multiple proteins, receptors, channels on the cell membrane. Some of these transporters need no energy, called the passive transporters. It may be channels or carriers, or the others need energy, and those who need energy usually are working against the concentration gradient, so they use the energy to reverse the concentration gradient. But the passive transport is with the concentration gradient, so no energy needed. We will concentrate on 2 pumps today, which is closely related to the water and sodium balance within the cell and outside the cell. So what is the mission? What is the target? If we discuss the compartments here, the intracellular space should contain 2/3 of the body water. So we need water to pass freely through the cell membrane, and the water can do this through a channel, passive channel, the aquaporine channel, and this channel is controlled by the antidiuretic hormone. This hormone concerned with sensing the high need of the body of the water, so it retain water. How can this hormone do this? By increase the expression of those channels. And here we need the sodium to be more in the extracellular space and less in the intracellular space, and the reverse to the potassium to be an intracellular. And this is against the concentration gradient, because the concentration, the sodium inside the cell is only 12 millimole per liter, outside is 145. So logically, if it's a pass through concentration gradient, it will pass from outside to inside, and we don't need this. So here, this is the sodium potassium pump. It's a sodium potassium ATPase. It's an active pump. You need ATP. Why you need ATP? Because it works against concentration gradient. What is the importance of this pump? We have multiple physiological rules of this pump, not only in the kidney or not only in the water control, but this is what we concentrate on today. But this is a very important pump in the action potential, in the neuronal transmission, and in the volume homeostasis. So those are the main two pumps we will try to discuss today. So what control the cell membrane? What control these actions? What control these channels? We have multi-systems to control this. But before we pass to the multi-system control, we will continue the facts. Those are the two simple the two simple channels we'd like to discuss or to highlight. Those one that I told you about, the aquaporine channels, those are the water channels. It's a pore mediated flow of water with rapid movement and temperature insensitive. The water can pass through lipid diffusion, but it's a slow movement and not carry the most effect on the water passage. Those are the aquaporins, and as we see, there is no energy needed. Those channels are called aquaporins, and this is the types of aquaporins. We have multiple aquaporins in the body, but we will concentrate on those who are present in the kidney, and especially on those under the effect of antidiuretic hormone in the kidney collecting tubules and the medullary collecting ducts. Why those are very important? Because those are the ones that sensitive for ADH or vasopressin, and any problem in these channels will cause loss of water, massive loss of water, in the form of nephrogenic diabetes incidence. And this is just a diagram of the type 1 aquaporin in the proximal tubule, type 2 in the distal tubule and collecting duct, type 3 also in the collecting duct, type 4 in the collecting duct. So those are the main sites of distribution, but as we see, we will try to concentrate on the aquaporin 2. This is channel number 1. Channel number 2, the active one. The active transport, as we see here, the process starts with fusion of the ATP with the channel, and this carry 3 places for sodium and 2 places for potassium. So the potassium enter the cell and the sodium get rid out of the cell. As I mentioned before, it's not only for water or volume control, it's for all the nerve control, action potential control, and the neuronal transmission. The systematic control. The systematic control is a harmony between 3 or 4 systems. The gut, which is the site of absorption and secretion of the sodium, water, and potassium and chloride. The endocrine system and the kidney. The endocrine system master all the process, and the kidney is the target organ, filtration, absorption, secretion, and the transmembrane shifts, which have some effect on the potassium and the water transformation more than the sodium transformation. But before pass to the control, the systematic control, I'd like to revise all the facts we discussed in the section 1. Number 1, human body is mostly water. Okay. Movement of water is regulated by the solutes passing through the cells. Okay. The water makes a larger percentage of body volume. It's not a pure water. It is in compartments. Osmoregulation is one of the very crucial hemostatic functions. Water is never transported actively, not need ATP. Under normal circumstances, a cell maintains its water volume at a constant level, but the composition of solutes will differ according to the solute transfer between the cell, inside the cell or outside the cell. To maintain the water balance, a cell control of the movement of the electrolytes to keep the total number of dissolved particles the same inside and outside. Some criteria of the cell and some terminology which is very important in getting the idea of the osmolarity and tonicity and how the water can pass between the compartments. We have the intracellular space, and this cell can this is a cell represent the intracellular space, and this is the cell membrane, and outside the cell membrane will be the extracellular space. The extracellular space is divided into interstitial space and the intravascular space, and the interstitial intravascular space is separated by a capillary membrane. The criteria of a capillary membrane differ from the criteria of the cell membrane. The capillary membrane can pass easily the water and the small solutes, but can't pass the large solutes like the protein or complex starch. The cell membrane is not permeable to solutes except if there is a concentration gradient between this compartment and the other, between the extracellular and intracellular compartment. Within the normal states, when the concentration of solutes here and the concentration of solutes here are the same, there is no passage of water between the two compartments. Okay? When this concentration differ, the water will be dragged to the site with higher solute concentration. Solute concentration, number of solutes within a volume of fluid, is the osmolarity. And the osmotic power of the solutes is the power of the solute to drag water. Okay? So if we have normal osmolarity state, our osmolarity is our human osmolarity is between 280 and 300 in the intracellular space, and then balance with the extracellular space. If any condition cause the osmolarity here, or the osmolarity or osmotic power here increased due to increased solutes that have a high osmotic power, so this compartment will drag the water from the cell and will cause cell shrinkage, and vice versa. If this compartment is hypotonic or of low osmolarity, the water will pass from it to the cell. The cell will drag the water and the cell will swell. Let's make some solidification. However, I told you about the cell, think about the brain cell. And the interstitium, think about the edema. And the intravascular compartment, think about which is called the effective circulatory volume. This is the blood plasma. And this is the volume which reach the major organs, the brain, the kidney, the liver. Those are the volumes that supply the organs. And this is the volume which the volume receptors and the osmoreceptors can detect. And this is a very important fact to know. The our body sense any change in body volume or any change on the osmolarity from this place. Okay, so you can have some cases with a problem in the blood volume in this intravascular space, because all the fluid from intravascular space escape to the interstitium and the cell, like in heart failure or liver cell failure or renal failure, nephrotic syndrome, because those pathologists have a problem in keeping the fluid in the intravascular space due to hypoalbuminemia or due to heart failure, congestion. There is all the fluid come to the interstitium. The blood here or the body here will sense there is hypovolemia or hypoosmolarity, so it will retain water and retain salt, and this is the problem of the liver cirrhosis or the heart failure or the massive nephrotic syndrome. So if we'd like to summarize, this membrane is guarded by the differences in oncotic and hydrostatic pressure. Those are the forces acting on this membrane, the capillary membrane. Because if I have a molecule here or solute here of high volume or high oncotic pressure or high hydrostatic pressure, it will affect the movement of water here. But in this membrane, there's a difference in tonicity. Tonicity is a relative term. Tonicity is the osmolarity of one space compared to another space. So if we take the body osmolarity as a reference in the cell, which is 300 or 280, this is the reference. Any osmolarity above this osmolarity will be hypertonic to this osmolarity. Any osmolarity less than this osmolarity will be hypotonic to this osmolarity. So the osmolarity is an absolute term, but tonicity is a relative term. So suppose that we have cell with normal tonicity and I put it in isotonic solution. So the number of particles and the concentration of particles inside the cell like outside the cell, so the water will not change or will not enter or exit from the cell. But if I put the same cell in hypotonic solution, like the concentration of solutes here is less than the cell, the osmotic power of the cell will drag water inside the cell, so the cell will swell. If it's the case in the brain, it will be brain edema. If it's a brain cell. In hypertonic saline, no. The solute around the cell has a higher concentration, higher osmolarity. It will suck the water from the cell and cause cell shrinkage. And this is again, if you think about a brain cell, it means brain herniation or osmotic demyelination. The last thing in the facts. A very important rule you should know in studying the topics with complex physiological or pathophysiological mechanism. You should simplify the knowledge and take a telescopic vision at first, then pass to the details, then at the end put the telescopic vision on the details. But you should know whatever the details of the channels and the hormones acting on the channels and the osmolarities and the concentration gradients and the pumps. We all know that the physiology of the kidney is quite complicated, but we should know which segment of the nephron is permeable or is responsible for the absorption of electrolytes and water. We should know this. Where sodium goes, the water follows, and I will explain this now. If you see here the filtrate contain sodium chloride, electrolytes and water. The proximal tubule is permeable to a site of reabsorption of the sodium and the water, but here to the water more, because as we see the urine osmolarity increase, So the sodium taken and the water taken, but the water taken more. If we pass late in the descending loop of Henle, the descending loop of Henle is not a site of sodium reabsorption. It's a site of water reabsorption. So the osmolarity of the urine will increase and increase because the salt is kept and the water is reabsorbed, kept in urine, and the water is reabsorbed to the circulation. Then till the ascending loop of Henle, the ascending loop of Henle is permeable to sodium reabsorption but not water reabsorption. So the dilution start again here. So as you see, there is a balance, and this is a counter current flow of water. So the osmolarity here is the same. Will enter the thick ascending loop of Henle, have this famous channel, which is the site of action of loop diuretics, sodium potassium 2 chloride. Again, it's a site of sodium reabsorption, but the water is still present. So there is a urine dilution till reaches the collecting duct, the site of water reabsorption, and the urine concentrated again, and this prevent the loss of water in the urine. This is the general idea. Let's pass now to the systematic control. So we fixed now some facts about the water. We know that the water is not actively transported. We know that the water is guarded by the solutes. The water follow the sodium. We have a passive water channel, aquaporin, as is controlled in the kidney in the collecting duct and distal tubule by the aquaporin, by the antidiuretic hormone vasopressin. We have an active transport pump present in many cells of the body, sodium potassium ATPase pump, which is responsible for the maintenance of action potential and the volume state. Okay, let's pass to the systematic control: the GIT, the endocrine system, the kidney, and the transmembrane shafts. Let's start with the GIT. GIT is the normal pathway for uptake of water solids from the body fluids. Water and the electrolyte balance in the body is regulated mainly by the kidney, but dysfunctional transport, like in diarrhea, will impact the whole body electrolytes. So the usual physiological, if the gut is okay, so its role is not crucial in the regulation. But if there is a diarrhea, it will affect the electrolytes. The GIT deal with around 10 liters of water per day, only excrete 1% of them, 100 ml. So there is a reabsorption, massive reabsorption of water, but the level, we should know that the level in the small intestine and large intestine. So the diseases which may hazard the small intestine and large intestine may cause a severe water depletion and may cause electrolyte depletion. Another fact you'd like to know about in the GIT: the water can move in either directions across the wall of small intestine depending on the osmotic gradient. But the ions sodium, potassium, calcium, and magnesium phosphate are actively transported. So here we have the water transport and we have the sodium potassium ATPase pump. Is the pump concerned with taking the sodium from the lumen, so dragging the water behind the sodium and preserve the body from loss of water. Again, as we said, the water is absorbed through passive osmosis. In cases with high concentration of unabsorbable solutes in the GIT lumen, the water can't be reabsorbed and thus cause an osmotic diarrhea. And this is a very important type of diarrhea. If the gut contains highly osmotic solute which is not absorbed, it will drag the water within the lumen and cause excessive diarrhea and fluid loss. And this can be used as a treatment for constipation or a purgative or preparation of endoscopy or preparation for colon surgery we can use this mechanism and osmotic diarrhea. And this is the same transport in what we discussed before. Two things I'd like to discuss in the context of GIT. The mechanism of CEREST is not related to the GIT only. It is a multi-process mechanism. But I'd like to mention it here. Why? Because in the patients with massive fluid loss, we should know why this patient can't replace this fluid with CEREST. Because it's a normal mechanism in our body. When the body volume decrease or the osmolarity of the plasma increase, there is an osmoreceptors in the hypothalamus which directly stimulate the ADH secretion and stimulate the CEREST center to make you hungry to water. And this is a very important mechanism to restore the water losses, like in diarrhea and or sweating. Or if the patient cannot restore this balance or this lost water, so maybe this mechanism is affected. I should know that the extremities of age, like the very young age and very old age, are liable to have defects in this mechanism, and I should think about this in managing the patient. The diarrhea. Diarrhea stool contain large amount of sodium and chloride, potassium and bicarbonate. Diarrhea is a very important cause of metabolic changes. I will discuss in the lecture dealing with the acid-base disturbances. The diarrhea will take some of the discussion. But now the loss of water, sodium and the potassium is an expected finding in the diarrhea. If the loss of water in excess of sodium, the sodium will appear as a concentration to be high, but it's not high. It is not just that the sodium loss is less than the water loss, and this is called hypertonic dehydration. So why hypertonic dehydration? Because of the dehydration, I lost water and I lost sodium, but I lost sodium less. So when I measure the serum sodium or the concentration of sodium will be normal or high. This is an hypertonic. What's the hypotonic dehydration? Hypotonic dehydration means that there is a sodium loss and water loss, but this patient is replaced with fluids with less sodium. So now the sodium concentration will drop. So this is a hypotonic dehydration. Potassium depletion is an expected finding in diarrhea. So this is the GIT. The main control is the endocrinal and renal control. So let's again speak simple, then pass to the depths. Simply, the hypothalamus and the adrenal glands detect the blood osmolarity. Kidneys and blood vessels detect the blood volume. How? The hypothalamus have osmoreceptors which detect sodium concentration. When there is a decrease, change in the sodium concentration, it will secrete ADH in the cases of hypernatremia. Hypernatremia means fluid loss, and this is a rule I'd like to solidify in this lecture. Hypernatremia, we measure not we don't measure the sodium itself, we measure the concentration of sodium. So if I found the concentration of sodium is high, so it means that the water is not sufficient for sodium. Okay, so it's a fluid problem, and the hyponatremia is an excess of fluid. Cells in the adrenal glands, since when sodium levels are low, aldosterone, it will secrete aldosterone. The kidneys and the blood vessels, in their blood vessels have a receptor which is the baroreceptors. Baroreceptors, since the change in the blood volume and stimulate the ADH secretions and the catecholamines. The juxtaglomerular apparatus in the kidney will secrete renin and angiotensin II. All of them will target renal water retention, sodium reabsorption, renin release, increased thirst, vasoconstriction. So we have major hormones. We have major hormones which act on this mechanism, which are the angiotensin II, aldosterone, and the antidiuretic hormone, and some minor hormones. We will not go in depth with those hormones, but we will mention the natriuretic peptides and the cortisol. The natriuretic peptides, from its name, it's atrial natriuretic peptide and prenatriuretic peptide. Both are secreted from the atria and the ventricles, and from their name, natriuresis, they directly increase the sodium excretion in urine. So they are against the action of those, the angiotensin II, aldosterone, and antidiuretic hormone. This is the sample. We will take some depths in this two major hormones, the renin angiotensin aldosterone system and the hypothalamo-pituitary adrenal axis. The renin angiotensin aldosterone system, a system stimulated by decrease in renal perfusion. So the juxtaglomerular apparatus will sense this and secrete renin. The renin help the conversion of angiotensinogen to angiotensin 1, and under the effect of the angiotensin converting enzyme, it will be converted to angiotensin 2, and the angiotensin 2 have a very important two actions: increase sympathetic activity, increase tubular reabsorption of sodium, chloride, and water, and excretion of potassium. And this angiotensin 2 will directly increase the aldosterone secretion, and the aldosterone do the same action, arteriolar vasoconstriction, and also increase the antidiuretic hormone secretion from the pituitary gland. All these are directed to increase the blood pressure by vasoconstriction of the arterioles and sodium water retention. The hypothalamo-pituitary adrenal axis is sensitive to the changes in the osmolarity. So when the osmolarity increase or the serum sodium increase, the osmoreceptor is stimulated, increase the ADH release, decrease and increase thirst. This will decrease urine water loss and increase water gain. The additional water will dilute the extracellular fluid, and the hemostat will sense that the osmolarity now is okay, so the mission is done. So those are the simple mechanism. Again, as I told you, in first, be simple. Be simple as much as you can. The renal handling of water. We should know, as we mentioned before, where are the sites with the water is taken, and where are the sites which the solutes are taken, and where are the sites with all of them taken but with different percentages. In the proximal convolute tubule, the solutes and the water. In the descending group of Henle, only water. In the ascending group of Henle, only solutes. In the collecting duct, solutes and water. And this is a very fruitful diagram with the hormones acting on the kidney. We have the angiotensin II acting on the proximal tubule more. We have the antidiuretic hormone or vasopressin in the collecting duct, distal tubule and the collecting duct. The aldosterone, distal nephron and collecting duct. The natriuretic peptides act on the distal nephron, distal convoluted tubule. I will leave this picture for you. Again, to go in depth with the renin angiotensin, the angiotensin II, as I mentioned, act on the proximal tubule, and the aldosterone act on the distal convoluted tubule and collecting duct, keeping the sodium and excreting potassium. So any blocker to this system or any aldosterone blocker will cause sodium loss and water loss and hypertension control, but hyperkalemia. Okay, this is the effect of renin-angiotensin system. In the collecting duct, the antidiuretic hormone will increase the expression of aquaporins, and this will reserve the water only. There is no relation to sodium, so as a free water absorption rather than sodium. An application of this, according to those aquaporins, as I mentioned before, if I have increased the expression of aquaporins in the any cause which increase the antidiuretic hormone. What are the causes which increase the antidiuretic hormone? It can be with no cause, like the syndrome of inappropriate antidiuretic hormone release. It will keep free water without sodium, so the effect will be hyponatremia. And if I measure the osmolarity of this serum, it will be hypotonic, because it's a water, excess water over the sodium. So this is one of the main causes of hypotonic hyponatremia. If the increase in ADH is due to false perception of high osmolarity or low blood volume, as I mentioned in the heart failure or liver cirrhosis, it will be a hyponatremia also, because the more fluid will be retained. But in this patient, there is no hypovolemia, it is a hypervolemic hyponatremia. If the aquaporin is not present or not sensitive to the ADH or not doing its action, so there is no urine concentration, and we will lose much more fluid or water in the urine, and this will cause the diabetes insipidus, and those are one of the main causes of hypernatremia. So we don't have now to study the causes of hyponatremia. We can think about the ADH state and think about. Sure, it's not only it's not only the only cause, but those are main categories of the cause, and those are the same as mentioned before. I'd like to you in this table to concentrate on the direct stimulus of the hormone secretion and the direct effect of the hormone secretion, the major effect, because we have major effects and minor effects, and we have major stimuli and minor stimuli. When study the major to be concentrated and to be fixed in your brain. Study the major stimulus and the major effect. The last systematic control is the transmembrane shifts. The transmembrane shifts, as I mentioned, from water and potassium. Transmembrane shift, water and potassium. Transmembrane of food, membrane shift of water. What does it mean? The transmembrane shift of water will be discussed in two examples. This example, if I have some patients with, it's usually this is a cellular compartment and this is a blood. If the normal regulation that the pancreas secrete insulin, and this insulin help the glucose to be entered through the cell. If I have a patient with poorly controlled diabetes and the glucose accumulate in the serum, don't find its way to the cell, so this glucose carry an osmotic and oncotic power. So this glucose will help to drag the water from the cell, because now in the intravascular space the oncotic pressure and the osmotic pressure is increased, so it will drag water, and this water will dilute the serum. Okay, and this, now the serum is diluted, have other solute rather than the sodium. So if I measure the sodium concentration, it will be low. But if I measure the serum osmolarity, it will be high due to the glucose. And this is a type of hyponatremia called hypertonic hyponatremia. And this is one of the pseudo-hyponatremias. Why pseudo? Because the problem is not in the sodium. The problem is in the glucose. So I measure the sodium. The sodium is low. Why? Because I don't measure the net sodium content. I measure the sodium concentration. Sodium concentration is low. Why? Because there is a drag of water and there is another solute. So the sodium will measure low. But the glucose have an oncotic or osmotic pressure, so the osmolarity will be high, so it's an hypertonic hypotone. The same in mannitol. It's another osmotic agent and will drag the water. This type of diuretics, osmotic diuretics, the same, so hypertonic hyponatremia. Okay, what can also disturb the measurement of sodium? If the patient have hyperlipidemia and hyperproteinemia, in this patient, when I take sample from the intravascular space, this is what we from the venous side, those will disturb the composition of the serum and will make me sense the sodium as a low sodium. And if I measure the osmolarity, those particles are not osmolarly active, so it will be normal osmolarity. So low sodium with normal osmolarity is a pseudo hyponatremia. It is a isotonic hyponatremia or pseudo hyponatremia. It's not the hyponatremia we'd like to have a problem in the sodium and like to treat. So now I just like to solidify that the hyponatremia I would like to find the cause or treat is the hypotonic hyponatremia. So we can exclude from the first the hypertonic hyponatremia and the pseudo hyponatremia. So measure the glucose and measure the lipids and proteins. This is very important to before dipping in the dilemma of hypotonic hyponatremia. The last thing is the transmembrane shift of potassium. The potassium carry a positive charge, and the H ion, which is the net product of the acid production in the body from all the vital processes, this outside the cell and this inside the cell. If I have excessive formation of H ion like in acidosis, the potassium will transmembrane shift to the outside the cell instead of the hydrogen to adjust the extracellular environment to be not too acidic, and because the too acidic media is not compatible with the functions of the body. So the acidemia will cause slight hyperkalemia, and the alkalemia will cause, and the vice versa, will cause hypokalemia. The second, that we have some hormones and some drugs which can affect the potassium shift from the extracellular space to intracellular space. We have the insulin and the beta 2 agonists can help the potassium to enter the cell, but for a temporary period, and this is one of the we can use this as a temporary treatment of life-threatening hyperkalemia that can cause a cardiac arrhythmia. So until I solve the problem and take diuretics and get rid of the potassium by any mean, I just need a momental solution to enter the potassium within the cell to prevent the hyperkalemic effect on the heart. This is a very important slide. I'd like to fix this in your brain. This slide is crucial. Those are the all mechanisms collected in one mechanism. So the GIT tract is a source of absorption of sodium, potassium, and water, and those are filtered to the kidney. The kidney will absorb the sodium, potassium, and water to some extent and excrete the other. Who help the kidney? The aldosterone and angiotensin 2 increase the sodium absorption by direct effect, and indirectly the water, because the water follows the sodium. The aldosterone inhibit the potassium reabsorption. The antidiuretic hormone stimulate only the water reabsorption, nothing on the sodium. The anti-natriuretic peptide and the pre-natriuretic peptide increase the sodium natriuresis or excretion directly, and indirectly the water excretion, because the water will drag behind the sodium, and the remaining will pass to the urine. The cortisol can make the action of aldosterone, but in excessive amounts. In excessive amounts of cortisol. The transmembrane shifts is a very important tool in the potassium and water, and we have the insulin and beta 2 agonists which can make the potassium enter decent. Now we will skip to the last section of the lecture, the applied aspects. We'd like to know how to think about the hyponatremia and hypernatremia, the fluids and the electrolyte changes with different types of diuretics, and the brain changes in sodium correction. This issue is now stable. We know now we measure the concentration of sodium, not the net amount of sodium, and the serum osmolarity is calculated from the sodium, blood sugar, and PON potassium. And because all in the normal conditions the potassium, sugar, and PON are not elevated, so we can consider that the osmolarity equal 2 by the sodium only. So in any thinking about the hyponatremia, we should know that we have an excess of water than the sodium. Maybe the two are decreased, but the decrease in sodium is more than the decrease in water. Hyponatremia equal, it's just an algebraic equation. We have the higher value and the lower value. So if the result of the equation is low, it means that the higher value is decreased or the lower value is increased. So in any case of measured sodium, low sodium, we will skip to measurement of osmolarity. So the hypertonic hypernatremia is not our issue, and the pseudo hyponatremia is not our issue. So I will exclude this first. And then if I see that there is a hypotonic hyponatremia, so the osmolarity is low and the sodium is low, I will skip to assess the volume status. Is it a hypovolemic? So there is a concomitant loss of sodium and water, and this can be the effect of diuretic, diarrhea, or vomiting. So the sodium and water loss can be from the GIT tract or the kidney. If it is hypervolemic, as I mentioned, the heart failure, cirrhosis, and advanced renal failure. The eovolemic, so to some extent the decrease in water due to water retention, we have some circumstances, but most important of them is the SIADH or the syndrome of inappropriate antidiuretic hormone secretion. So the simple how to think in hyponatremia, we have to measure serum osmolarity, normal, low, elevated. If low, so hypervolemic, hypovolemic, eovolemic. If it's hypovolemic, I will check urinary sodium. If the sodium loss will be from the kidney or effect of diuretic or adrenal insufficiency or cerebral salt twisting, it will be a high urinary sodium. If the patient is below 15, so there's other source of water sodium loss in the GIT tract or PERN or pancreatitis or something like that. So this algorithm, which is more typically both the cerebral salt twisting, the difference between cerebral salt twisting and the SIADH is the cerebral salt twisting is slightly hypovolemic. And this is how to think in hyponatremia. Another thinking, if it's related to the ADH, I can it's inappropriate ADH secretion, okay, or some endocrinopathies which increase the ADH like hypothyroidism and adrenal insufficiency, maybe okay, or some problems which cause a false sensation of decreased blood pressure or effective circulatory volume like in heart failure or cirrhosis, or ADH appropriately suppressed like in advanced renal failure. This is the application number one, the hyponatremia. Application number two, the diuretics. we should know a very targeted information about each diuretic. The main site of action, and this main site of action determine the power of the diuretic and determine the expected water, sodium and electrolyte loss by this diuretic. I will not pass to discuss now or in this lecture the metabolic effect or the effect on calcium homeostasis, because I will mention it in a separate video, but I will concentrate on the sodium, potassium and water. The loop diuretic will act on this very famous channel, sodium potassium 2 chloride channel in the loop of Henle, in the ascending loop of Henle, and this channel is concerned with 25% of filtered sodium reabsorption. So it's a very strong channel. So block of this channel through competition with the chloride will make forced or powerful diuresis, because we will lose 25% of the power of the sodium reabsorption and consequently the water and the potassium. Another mechanism, if the sodium is reaching the collecting duct in a high concentration, there is a mechanism in the collecting duct to secrete potassium in favor of sodium, and this will cause hypokalemia. So the loop diuretic is a powerful diuretic, will cause loss of sodium and water and cause hypokalemia. The thiazide diuretic acting on this channel in the distal convoluted tubule with only 10% effect on the filtered sodium, and in the potassium will make the same effect, so loss of sodium, water and hypokalemia. The potassium sparing diuretics act by different mechanisms. We have two types, the aldosterone blockers and the epithelial sodium channel blockers. Both are in the collecting duct, and this is a very small amount of filtered sodium, so the diuretic effect may be less. So they used in combination with other diuretics, not an only drug. And the difference that those diuretics will keep the potassium. We agree that the aldosterone action is to cause hypokalemia. So if I antagonize the action, it will cause hyperkalemia. So this will cause loss of sodium and water, but hyperkalemia. The carbonic anhydrase inhibitors acting on the carbonic anhydrase enzyme in the proximal convoluted tubule, so lead to sodium and bicarbonate loss, and this will cause hyponatremia and acidosis, but it's 60% of the filtered sodium and 85 of the sodium bicarb, but there is a tolerance from the action, so it's not so powerful. We can't rely on it on a regular basis, and also cause hypokalemia. The osmotic diuretics, mannitol, we discussed the mannitol, it will cause free loss of water, reduce the intracellular volume and the hypernatremia. Why? Because it will not affect the sodium reabsorption, but just by its osmotic power in the urine will drag the water. The hypernatremia is another issue. If I think about the hypernatremia, what's the problem of hypernatremia? Hypernatremia equals fluid loss. So what's the cause of fluid loss and why the patient is not gaining the fluid loss? So I should check for the thirst and all the steps of thirst, the sensors of thirst, the sensors of osmoreceptors and volume receptors, then the brain center, then the GIT tract, then. But if we exclude this, we have loss of water. Loss of water may be through GIT losses or insensible losses, I should make, or through the urine, And the through urine, why I lose the water through urine due to inadequate ATH, or if there is an osmotic diuretic. So a list of causes, okay, a list of causes, but the idea is very simple. We have on the other side, which is not common, is to have sodium overload, not loss of water, sodium overload by excessive administration of saline, or transcellular movement of water can be in the vigorous exercise or seizures. The last thing is the brain effects from sodium concentration. The brain is the most osmotically sensitive organ. Brain is the most osmotically sensitive organ. And as I mentioned you in the beginning of the lecture, that suppose that the cell of the intracellular space is a brain cell. So if the osmolarity changes in the extracellular space, the brain cell will react with swelling or with shrinkage. And this is a very important issue we should know in the correction of hyponatremia and hypernatremia. So if I am treating a patient with hyponatremia, it means that the extracellular osmolarity is less than the intracellular osmolarity, and this will drag the fluid to the brain cell, and the brain cell will swell. So if I am treating the patient with solutions contain sodium to treat the hyponatremia, so if I drag the water from the brain cell in a rapid way, it will cause osmotic demyelination syndrome or central pontine myelolysis. And the reverse in the hypernatremia, the cell is shrinked. If I infuse the patient with hypotonic fluid, so the patient will give cerebral edema. And this is why they put a rule for this: not correct the sodium more than 8 to 9 milli equivalent per liter. And you now in the first 24 hours, and you now know why, because I am not just adding sodium. No, I am dealing with the brain fluid, with the brain cell size, which is not adaptable in the rapid changes. It needs time to change, so need 48 hours, 72 hours to take the normal size again. At the end, I apologize for the long time of the lecture, but I insist to say all the applied aspects beside the normal physiology or the physiological background, because we should study like this. Thank you and goodbye.